I'm joined today with Dr. Nigel Plummer, an expert in the field of probiotic research and innovation. Dr. Plummer, what goes into making a good probiotic? What are the mechanisms behind a clinically effective good probiotic? How do we make it?
Well, it's a complicated manufacturing process overall. And how we start, let me just go through the whole process briefly, even though it is complicated. So what we start off with is a little vial that contains 5 mils of our probiotic strain. And we then put that into a series of inoculum vessels, and we basically grow it almost like in a soup. So we take the 5 mils and we put that into a 5-litre flask, and we turn that 5 mil culture into 5 litres, and then we put the 5 litres into 100 litres, and then we put the 100 litres into 1,000 litres, and then 1,000 litres goes into 10,000 litres. So in the end of this fermentation process, we have maybe 10,000 litres of this broth, and that contains the microorganism, the probiotic organism. Then what we have to do, because it's in 10,000 litres, it's in a dilute form. So then we have to basically concentrate it. And we can do that by 2 different means. We can either do it by a filtering method, or we can centrifuge it. Either way, what we end up with from that 10,000 litres is about 200 litres of a concentrate. And this concentrate looks like double cream. So it looks like double cream, and that's and then what we then have to do at that point, now we've concentrated it, because you see probiotics in a freeze-dried or in a capsule form. And in that form, what it contains is a freeze-dried powder. So we've got to take that concentrate that looks like double cream, and we've got to turn it into a freeze-dried powder. Right. But freeze-drying, this is a critical point. Freeze-drying is actually, although it's a very gentle drying process, it's still harsh on a living organism. And there are 2 areas where you can cause damage. One is the freezing process, and one is the drying process. So freezing, when you freeze, obviously you produce crystals of ice. And sometimes if you freeze it in the wrong way, then you can produce these large crystals of ice, and they can pierce the cell membrane, for instance, and that can kill the bacteria during the freezing process. So what we do during that is we add salts to help the freezing process and to decrease the ice crystal size. And these salts might be saline, for instance, or a phosphate salt. So a phosphate buffered saline is the type of thing that we would use to decrease crystal size during the freezing process. So once we've got that done, then we've got the drying process too. And here, when we dry, we freeze dry. So we freeze dry at minus 30 degrees C. So it sounds crazy, but we actually dry at minus 30 degrees C. And think about it like this, that you have the frozen microorganism, and we're almost vacuuming out the water molecules. And when we do that, the 3-dimensional structure, because water molecules hold the 3-dimensional structure of the proteins in the cell, and they hold them in that structure. When we take water out, those 3-dimensional protein structures collapse, and they lose functionality. So again, the bacteria dies. So what we've found is that there are certain sugars, sucrose and trehalose, that are perfect, that when we start to take water out from the drying cell, that the sucrose and trehalose molecules, they fit back in, almost like scaffolding. So as we're taking water out, the sucrose and the trehalase molecules fit in, and they take the place of the sugar, and they're almost like scaffolding, holding the 3-dimensional structure of the protein in its original form. And then we can then dry it. We've taken all of the water out, and we've replaced it, as I say, with this sugar scaffolding, if you like. So sugar works very, very well, trehalose, sucrose particularly. And then when the organism, when the probiotic is reconstituted again, when there's water available, what happens, the water kind of comes in, it comes gushing in, and as it does that, the sugars are released from the 3-dimensional structure, and the water replaces the sugar, and it's almost like scaffolding being replaced by
Dismantled.
scaffolding.
Right.
And so it works fantastically well. And we've understood that now over the last 4 or 5 years, and it's made a major difference in terms of survival of probiotics during this whole manufacturing process. So saline and phosphate buffered saline, particularly important in terms of decrease in the ice crystal size, protecting the cells, and then trehalose and sucrose as sugars that end up as being scaffolding in the three-dimensional structure of the proteins, again protecting the cells from dying during the drying process. And that's basically how we do it. So then we come out of the freeze dryer comes this concentrated powder, and that's what goes into the freeze dry capsules.
Thank you, Dr. Plummer. That was an excellent technical review of what actually makes a good, clinically effective probiotic.